Yeah, okay. Okay, so today we are here to hear Wen-Yun Yang's thesis defense. Wen-Yun's done a lot of work in our lab on, he's going to be mostly talking about his work on spatial ancestry, but Wen-Yun has been the resident optimization person in our lab, and so he's actually solved every hard optimization problem in the lab in the last 5 years. So exactly how we're going to function without him is not clear. It's been great having Wen-Yun. Wen-Yun's brought a lot of spirit, and he always has a very different perspective on things. One of the few things I can say is that you haven't really experienced who he is until you visit him in China, which is a completely different environment. So it's been really great. He's been in the lab for 5 years, and he made many contributions he'll talk about today.
Okay. Thanks, Lar, for the introduction. So welcome everyone to to my defense. So today I'm going to talk about my thesis. So the title is Efficient Probabilistic Model-Based Approaches for Analysis of Human Genomic Data. So the title is a little bit vague because we want to include, I want to include a couple of projects. So in previous couple of years, I have been working on a couple of projects. So they can be categorized into 3 main areas. So the first one is the spatial ancestry analysis, spatial genetics. The second one is sequencing related topics, and the third one is genome-wide association study related topics. So today I probably don't have enough time to cover all of them. So I will cover the 4 main projects I have been working on. So I'll 3 of them from spatial genetics and 1 from the sequencing related topics. So before getting into the project, first let me give some background knowledge, and so that for people who are not familiar with this genetic terms, so we can get familiar with them. So for all the topic I'm going to talk about, we are going to study single nucleotide polymorphism. So for short, we call it SNP. So here's some example. We have 5 individuals, and each individual have their chromosome. And the chromosome can be understood as like a chain of nucleotides. And for most of nucleotides, they are the same across all people. But there exists some nucleotides. Different people show different variations, so we call it SNP. And we know that most common SNP have only 2 possible alleles. So in this case, we have A and G, but not C and T in that SNP. Okay, we also need to know what is the genotype and haplotype, because I'm going to use this all over this talk. So just take this example. I have my paternal chromosome and the maternal chromosome. They are in blue and green color. So when we talk about genotype, it means the set of 2 alleles at each SNP. So basically, also we know some homozygous SNP and heterozygous SNP, because if they are, my paternal and maternal allele are different, so we call it heterozygous. For the genotype, when we say at the first SNP, my genotype would be 2A. So, and the second one, just the set of my paternal and maternal allele, so it's 2C, and the third one is 2G. So actually, because they are the same, we can just call them homozygous A, homozygous C, or homozygous G. And we have the heterozygous here. So this genotype will be AC, but when we talk about genotype, we don't really know which allele from the paternal and which allele from the maternal. So without the color, we call it like a heterozygous AC. So on and so forth. So my genotype for these 2 chromosomes will be this way. So the haplotype will be easy. So it's just a set of alleles in 1 chromosome. So we have the first haplotype will be this way. Second haplotype will be just the first line and the second line will be my 2 haplotypes. Okay. So now let's move to my first project, spatial ancestry analysis. So we call it, for short, we call it SPA. Okay, in this project, from the name, we can know we want to do the ancestry inference in the spatial, basically. Okay, so first of all, modeling the population structure is a fundamental topic in human genetics. In this project, we are interested in the spatial structure of genetic variation. So in particularly, we are interested in using a simple but useful function to model the spatial distribution of the alleles. So in this example, we have like 3 genotypes. And from the map, we can see there are more alleles A in the West than the East Europe. So in this case, we would like to model this pattern by using a function in the right. So that one will give the allele frequency of A over the Europe. And this is the intuition, but mathematically, we model this pattern using a logistic function, so which is given here. So there is one variable here, which is x. So x is a 2-dimensional vector, which stands for the location in the map. So latitude or longitude is, and we also have 2 parameters in this function, which are a and b. So for different parameter a and b, we basically can model different allele frequency pattern in the space. So for example, here, if the magnitude of a is very small, so most likely we will get like allele frequency slope function, which are almost flat across all Europe. And if the magnitude of A become a little bit bigger, we will get like a gradual change allele frequency over the Europe. And if the magnitude of A is very big, we probably get a very sharp change of the allele frequency happen somewhere in the Europe. Okay, so now the idea is that given the allele frequency slope function for every SNP, and assume we have like hundreds of thousands SNPs, and for every SNP we have their allele frequency pattern in the Europe, for example. And for example, here we have the allele frequency of A, allele frequency of T, and allele frequency of C for 3 SNP. And then when we are given a new guy's genotype, we are actually able to predict where the new guy is from Europe, like somewhere in the Europe. So basically, for example, the first SNP might not tell us a lot of information because the allele frequency pattern is so low, so flat. So there, and everywhere you can have allele A. And for SNP 2, and because here you have a 1T, so it's more likely this new individual from the Middle Europe or the West Europe. And SNP 3 will tell us a lot of information because only in the West or like Middle Europe you can get allele C. So this one will have a lot of information. So basically, by combining all the information for millions of SNPs, we can actually predict where the guy is from. And mathematically, we solve this problem using convex optimization techniques. So it can be efficiently solved. So we apply this method to the POPRES data set, so which has 1,300 individuals and almost 500,000 SNPs. So every individual here we used, it has 4 grandparents from the same country. So they are not mixed individual. So this data set has already been analyzed by John November in 2008 using the principal component analysis method. Okay. So this is the cross-validation result of our method. So we use 10-fold cross-validation. So it means that every time we use 9-fold of individual to fit the allele frequency slope function, then we predict the location for the rest 1-fold individual. So we do this 10 times, then we have the prediction for every individual. So left is the result. So if we compare with Europe geography, we can see there are very similar pattern. Okay.
Did you try? Did you try this naive approach where, let's say, you have 9-fold individuals with their known location, and then you come up with a new individual, just assign it to the location of the closest in terms of like having distance?
Yeah, I think before this project, at the first year, I tried different methods. I tried probably 4 other methods, but it's not finding the most close one. It might find like the 10 most close one. That's what actually works. I mean, not as well as this, but I can see it works. Yeah. Okay. So far we learned basically 2 directions. One is if we have the allele frequency slope function, then we can predict the individual location. And then reversely, if we have the individual location, we can fit the allele frequency slope function. But now the question is, what if we have a data set without any individual location information? Then basically we only have the genotype information. So what can we do from there? And the answer is that we can just randomly initialize the individual location, like the iteration 1 here. Then we fit the allele frequency slope function. Then we predict the individual location again. Then we do this alternatively until the mapping gets converged. So which after maybe around 10 iterations, we can see the mapping is already get converged, and it's very similar to the Europe map. And if we compare to the principal component analysis results, our method is a little bit better because the population gets separated better there. So one typical drawback of the principal component analysis is for the mixed individual, it doesn't work well. The reason is, for example, if we have a mixed individual, the father from Ireland and the mother from Spain, so the PC will just place that guy between the paternal and maternal location, so which might be in the ocean. So it doesn't make sense at all. So in this case, our method basically can rescue this guy by splitting the prediction into 2 origins. So basically we use 2 locations to represent the mixed individual, so they can be in Ireland and Spain, not in the ocean. Okay. So we apply this data to the simulated individuals, and we get around 50% parental country origin correctly predicted. So actually we have 33 countries. So basically we want to pick 2 countries from the 33 countries, so we get a 50% accuracy. Okay. Now the question is, because we just did the experiments on the Europe data set, now the question is if we want to apply this method to the whole world population, there will be some problem, because if we want to fit the allele frequency slope function here, basically Alaska and Siberia, they will have a very different allele frequency, right? Because we're using a slope function to model this. And however, this is not true, because the Alaska and Siberia, they're actually close to each other in the other side of the world. So basically, we cannot use slope function here. We need to model everything using in a global, basically. So by a simple extension of the previous logistic function, we can actually model different allele frequency patterns in a globe. So like we can model like almost no change of allele frequency and gradual change of allele frequency from the south pole to north pole, and sharp change of allele frequency from west to east. So by modeling the allele frequency in this way and applying the similar method as previously, we randomly initialize the individual location in a globe. Then we fit the slope function, then we place them again. We do this alternatively until we get converged. Then we can see here, like after 10 iterations, and basically if we compare the final mapping with the true globe mapping, so we can see it's similar, but not that much, not as well as the Europe map. So we can see the East Asia, the Central South Asia, Middle East, Europe, Africa, Oceania. So they kind of follow the same topology here. And also, if we rotate this globe, we can actually find the Native America is exactly in the other side of the world.
How many parameters do you add to the model to do this?
How many what?
How many new parameters do you add to the model?
One more. Actually, just one more dimension to model the three-dimension thing. And we also have a constraint to place the individual because the individual has to be in a globe. It cannot be randomly. So basically one more parameter, but one more constraint.
So one more parameter per snip.
Right, one more parameter per snip, yeah. Okay, so one more thing that our model is able to do is because we are explicitly modeling the allele frequency, so we are able to detect some possible selection using the allele frequency function. For example, here, if there is a very sharp change of allele frequency in the Europe, so it might suggest some possible selection. So if we do a genome-wide scan of all the SNPs I have, so basically we can identify the lactase region has the strongest signal of the, basically almost every SNP there have very sharp allele frequency change. Allele frequency change. So basically here, this is the represent how steep the allele frequency pattern is. So we can actually visually see this allele frequency change. So this is basically in the Europe, like this is north direction, this is east. So we can see here in the, this is some SNP in the lactase region. So basically in the southeast Europe, we have very low frequency, and in the northwest, we have a very high allele frequency. So there is a clear pattern there. But if you compare with the typical gene, there's almost no pattern. So we apply this result to like all the SNPs, and this is a histogram of the steepness of every SNP, and basically this direction gives you the deeper gradient. So the lactase is like has very strong signal, and we also have other genes have a strong signal, which is like HLA, OCA2, FOXP2, and LR1B. So we also know that OCA2 is related with the hair color, eye color, and FOXP2 related with some language ability. Okay, so this project has been done last year, and the paper got published, and we have a software available online. So one typical use of this software is if you have your 23andMe data, you can run our method to get a prediction of the in the Google map, basically. So far we have 2,000 downloads and 20,000 page view. Okay, so now let's talk about the second project I have been working on, which is SPA for the mixed individual. So you might be wondering why we are doing this as a mixed individual again. So let me explain why. So in the previous SPA project, the first thing we did is to predict the individual location, but that's only one origin for that individual. So the basic assumption is that we assume every allele from the 2 chromosomes of the given individual is from one location. So this is a basic assumption. And we did a simple extension for the parental localization. So we predicted 2 locations. However, there we also made a basic assumption. Basically, 1 chromosome is from the father and 1 chromosome from the mother. And there is no haplotype mixing. So in here, like the first chromosome is from the England, second one from Italy. And we assume this haplotype, the first one can only from England. There is no mixing. So this actually just a simple extension, we can do that. However, these 2 methods are not applicable to much more complicated ancestry analysis case. So that's what we want to solve in this project. So in here, the assumption is the haplotype can be mixed, and actually we want to predict 2 locations, but the haplotype can be mixed, and we don't know which chunk from which location, and we don't know the boundary of those chunks. So all these are unknown. What we have is only genotype data and a bunch of allele frequency slope function for all SNP. So So this project, the target is we want to find the two locations as well as the genome chunks, and we want to assign those genome chunks to the two locations. So let me just give the intuition of how the algorithm works. So remember, we have two tasks in this project. The first one, estimate the location, for example, two locations. And we also need to account for the uncertainty of the ancestry assignments because we don't know which chunk from which location and we don't know the boundary. So in this case, first we randomly initialize the location as previously. Then we, this basically gives the hypertype. This is the ancestry assignments. So the color corresponds to location. So given the two locations, we are actually able to guess which chunk from which location. This actually can be solved by the forward-backward algorithm in hidden Markov model. Okay, then basically we iterate between these two things. So we update the location based on this ancestry assignment. Then we update this ancestry assignment, and we do this alternatively until it gets converged. So this is the intuition. But in reality, what we did here is the whole thing is actually a probabilistic model, and the way we solve this is the EM algorithm, so expectation maximization algorithm. So this step, the first step is actually the M step in the EM algorithm, and the second step is the E step in the EM algorithm. And this one can be solved by the Newton method. This one can be solved by the forward-backward algorithm in the hidden Markov model. So again, we apply this method to the POPRES dataset, but to different individuals in the POPRES dataset, because there exists 400 admixture individuals in the POPRES dataset, but they has never been analyzed because in all the previous study, we just remove all the mixed individuals. So this is the first time we analyze those self-reported admixed individuals. So this is the result. So the first row is a simulation result. The second row is for the real self-reported mixed individual. So different columns is different country pairs. So here the French Swiss and the British French and the British Swiss. So just by average, we get around 500 kilometer to 400 kilometer to 500 kilometer error rate between our predicted location and the true country center. So this paper now is under review, and the software will be available after the publication, basically. Okay, so now let's move to the spatial-aware hypertype copy model. So we call it SPA-HMM. So this is done actually in the previous couple months. So first of all, what is a hypertype copy model? So the basic idea here is the target hypertype can be modeled as a mosaic copy from the reference hypertypes. So in this case, for example, this is maybe one of my hypertype, and we have a bunch of reference hypertypes here. So if we think that my hypertype is a mosaic from the reference hypertype, then basically we can find a path in the reference hypertype, and this basically stands for which hypertype my hypertype is copied from. So in this way, we can think my hypertype is copied from this chunk and that chunk. So there are typical application of the hypertype copy model is genotype imputation. So in this way, we can infer the unobserved genotype or hypertype. So here the example, I have two missing alleles for which maybe we just don't have that data. But however, we have some other alleles is observed. So by using the same path from the previous slides, we can actually find the path in the reference, then copy them back to my hypertype. So in this case, if we make this copy, actually, we can also make a guess that this allele might just be the same as this chunk from the reference. so it should be like a T or C. So this is like the basic idea of the genotype imputation. Okay, so yeah, so there is a one assumption, implicit assumption of this traditional hypertype copy model is that my hypertype has the same probability to copy from all the reference hypertypes. So so there's basically I treat all the reference hypertype equally. So this makes sense, but it's not true for all the data. So that's the motivation we propose this spatial-aware hypertype copy model. So the idea here is we think the hypertype should be copied from is more likely to copy from nearby hypertypes. So for example here, this is my hometown where I come from, and we collect all the hypertypes all over the map. And some hypertypes are close to me, and some hypertypes are far from me. And we use the color to represent how far the hypertypes are from me. So the darker color means they are closer, and the lighter color means they are far from me. So in this case, if we think that my hypertype, so it's actually very natural to assume that my hypertype is more similar to the hypertype nearby me. So it's very likely that my hypertype should be a path like very close, very in the upper side basically. So it should be like this instead of some random path in the bottom. So yeah, so this my hypertype should be more darker basically. Okay, so now I just showed the intuition, but here we want to model this mathematically using a more rigorous way for this. So we basically encode the spatial awareness into the transition probability. So for example here, because we have a switch between hypertypes happens here, so we must define some transition probability. Basically the whole thing is a hidden Markov model, so here we define the transition probability. So the first transition probability is just stay the same chromosome. So the probability is 1 minus theta, and theta will be the recombination probability. So 1 minus theta means there is no recombination. So this is the transition probability for here. And we also need to define the transition probability for the switch between hypertypes. So here we saw a big term there. I will explain one by one. So the theta basically means the recombination probability. So actually the theta is already very small because we don't want the recombination happen too much. And there are big terms there, basically represent the spatial awareness. So we define a phi xi as the distance between x and xi. So x is the target hypertype location, and xi represents the reference hypertype location. So the phi xi represents the location between me and the reference hypertype. And we have a term here, lambda. So this one is the important parameter we are going to use. So this represents the spatial effect parameter. So we can know that if this lambda equal to 0, actually the spatial-aware hypertype copy model will be reduced to a traditional model, because the distance won't play any role there, because if lambda equal to 0. And if lambda is very big, so I probably can only copy from very, very nearby hypertype, like only nearby 2 or 3. And the parameter, the variable z is just a normalization factor to make everything to be a probability, so we can ignore it for now. Okay. So we apply this model to the 1,000 genome data set, where we have 14 populations. They are all sequencing data, and we apply to 150 SNPs from the chromosome, 1,000 SNPs from the chromosome 22, and we use around 2% SNP to impute the rest 19% SNPs. And here it shows where the population from. There we show the PCA mapping of all the populations. Yeah, first of all, I want to show that our model has a much better likelihood than the traditional model. So this x-axis represents the population, and the y-axis represents the log likelihood ratio between the spatial-aware model and the traditional model. So here, basically, we can very obvious the spatial-aware model has a much better fit to the data than the traditional model because every population shows better likelihood. Here is the imputation accuracy improvement. So we divided it into two panels. The left one is for the low frequency SNP, which is like 1% to 5% allele frequency, and common SNP is larger than 5%. So the x-axis here represents the spatial effect parameter lambda, so it shows the lambda value, and the y-axis represents the relative improvement from the traditional model. So this black dashed line represents the traditional model, and because if we know the spatial effect parameter lambda equal to 0, then our model will be reduced to the traditional model. So here basically they are the same. Then when the spatial effect parameter lambda increasing, we can see the imputation performance get increased. Basically under a reasonable value of the spatial effect parameter lambda, we can get for rare variant we get 5% improvement, and for the common one we get around 2% improvement. So actually here I just show the pattern of the improvement, but in the paper we actually develop an algorithm to estimate the optimal lambda value to get the best performance of imputation.
For the American individuals, are you using the ASW and CLM in those populations? And is that one doing so poorly because those are the populations which are fairly admixed themselves?
You mean here?
Yeah, because as you go more spatially aware, you're pulling more from those populations than you are from the populations that might have admixed into them.
Yeah.
So if you were to do like local ancestry on them, would you expect that, and then do a local ancestry aware haplotype version, would you expect that to rise?
I think the reason for the Native American performance low is just about some of them mixed, and the other reason might because they are not that similar as other population, because we collect the American Native American populations like some are in South America and some are in North America.
Okay.
Yeah. Okay, this paper is also under review. Okay. So now I'll talk about the last project, which is the Haplotype Inference using sequencing read and reference. So for short, we call it HARSH. Okay, so first of all, what is the Haplotype Inference problem? So the reason we have this problem is because current high throughput cost-effective sequencing technology only gave us the genotype but not the haplotypes. So basically the current technology only tell me that my genetic information would be all the genotypes. However, we don't know the haplotype from the current technology, and there exists a lot of possible haplotypes. For example, the haplotype might be this or might be this. As long as the alleles are consistent between the haplotype and the genotype, they are possible haplotypes. So we can actually easily to calculate there are a total of 2 to the n different solutions. So the Haplotype inference problem basically is we want to find the correct solution from these 2 to the n different solutions. So there exists a bunch of methods for the Haplotype inference. One approach is the Haplotype phasing using reference. So for example, here given my genotype information, and then we collect a lot of reference haplotypes. Then we want to basically reconstruct my haplotype from all those reference haplotypes. So what happens is also here we don't care about those homozygous SNP because they doesn't matter for the haplotype inference because either from the father or from the mother, it's done the change of the haplotype. So we only consider the heterozygous SNP here. So the intuition for the haplotype phasing from reference is to leverage the strong correlation between nearby SNPs. So in this case, let's first look at the first two SNPs. Then we will see, okay, if the first SNP is A, then the second SNP is very likely to be C because there's a strong pattern in the reference panel. So okay, we might make the guess of my haplotype will follow the same pattern as the reference panel. And if we look at the second and the third SNP, we might observe the pattern that the C might be followed by G and the G will be followed by T. Okay, then we make this guess. So basically by we repeatedly doing this, then we get my haplotype. Okay, but this, I just show the intuition, but this is not how the real algorithm works. So in reality, when we do the haplotype phasing, it's actually the same idea as the haplotype copy model. Basically, we find two paths. Instead of one path, we find two paths in the reference panel, such that my genotype can be recovered from the two paths here. Yeah, that's how the haplotype phasing works. So there's another approach, basically from the sequencing read, because the sequencing read have some partial haplotype information. Because the current sequencing technology only gives the complete haplotype, but it's able to give some partial haplotype. So, for example, we have a sequencing machine, and we have a lot of read, and we assume the unique mapping to the genome. Then we are able to see that for those heterozygous SNP, this read actually tells us a little bit information that the CGT should be in one haplotype, and this haplotype will tell us, this read will tell us a lot, another information for the paternal. So what happens is we want to use those read information to get back my haplotype. So actually this is what really happens, because we, in reality, we don't, from the sequencing read, we don't really know their colors. So basically we don't really know which read is from which haplotype, but we do have this CGT information show that they should from one haplotype. So, okay, so now the question is we want to find two haplotypes to best represent the read. So we also have some sequencing error here because there exists 1% sequencing error, so we changed that alleles. Okay, so now let's consider one possible phase solution. And then what happens is that we, for every read, we can assign the read to the haplotype such that the sequencing error is minimized. So in this case, we can give them colors. The color basically represent the guess of this read from the which haplotype. Then we can find, okay, there are 5 errors because if we assign this read to the second haplotype, the two alleles are different. And basically the total we have 5 errors. Okay, so then let's consider another possible case. If we phase the data in this way, then we repeat the procedure. Okay, then we assign all the read to the most to the haplotype that minimize the error. Okay, then we find, okay, there are only 2 errors. So in this case, between the possible phase 2 and the possible phase 1, we will pick the possible phase 2 because in the possible phase 2, we can minimize the sequencing error when we assign the read to the haplotypes. Also, the target of this haplotype assembly is to find these 2 haplotypes such that the sequencing error is minimized. Okay, so far we learned 2 approaches. First one is haplotype phasing. from the reference to the hypertype, and the second one from the sequence read to the hypertype. So however, these two methods, they have their own advantage, but also have their own disadvantage. The disadvantage is that they cannot use the information from the other method. So this one, it will ignore the information in the sequence read, and that one will ignore the information in the reference hypertypes because the method nature, they cannot do this. So basically, we want to combine these two methods. Basically, we want to use both information together to do a better job here. Okay, so the overview of our method is from reference panel and from the sequence read. We want to recover my hypertype. So we model this problem using a probabilistic model. So here we have, let me explain them one by one. So we have three types of variables. The first one is represent the reference. The second, the variable H represents the hypertype, and the variable R represents the read. So they are all random variables, and we have some meaning here. So this S variable, it can be, if we have 100 reference hypertype, their possible value might be from 1 to 100. It represents that from which reference hypertype this SNP is copied from. So it represents like which reference hypertype. The variable H will represent just the two hypertypes because we don't know them. They are completely unknown to us. So they are random. They are binary variable to represent that. And for this variable R, it represents the origin of that read. Basically, because we don't really know every read, it's from the first hypertype or the second hypertype. So we actually don't know their color. So this variable is a binary variable to represent whether the first one or the second one represent this uncertainty, basically. And the second read also represents this. And so the whole problem here is for every configuration of all the random variables, we can actually have a likelihood function. Likelihood function will be defined using all those edges. And the target is we want to find the configuration of all random variables such that the total likelihood is maximized. So this problem is a little bit hard. I tried a different method, and so we know that the exact inference is NP hard. So we have to do a lot of approximation method. I tried different deterministic method, but they don't work well because partially might because there are too many loops in the graph. And Dan He in our lab previously, he developed an existing dynamic programming method. It's very impractical in time and memory. So eventually, actually, we use a keep sampling based method, and which actually works the best among our algorithms. So we did experiments using the HapMap data, and we tried different settings for the experiment. Here's the result. We compare our method hash to the assembly method and the BIGO and impute. The BIGO and impute are the hypertype facing method. Assembly is the hypertype assembly method. So here we show different setting. One is a different coverage, one is a different rate length. And this y-axis represents the error rate. So we can observe that in the different setting, our method works the best. And yeah, that's it. Oh. Yeah. Yes, because input and BIGO, they cannot use the sequencing as input. But in this experiment, we use the basically BIGO input hash use the same reference panel, But for hash, we also use sequencing read.
Okay, so up here on the same individual, same set of individuals.
Yeah.
So I wonder, like, when if you change, like, to a genotype to a better lower error rate genotype on the vector genotype, would it help to improve there?
Sorry, what's the question?
So I don't know whether, like, because you just take their results
Mm-hmm.
from, like, learning on some genotypes,
Uh,
right? Or you copy the genes from the
I think we use the likelihood from the sequencing rate, because if we have the sequencing rate, then we for every SNP we can compute like how many read have a A, how many read have a C. Then we can assume the binomial distribution. We have a likelihood for that genotype, and when I run this experiment, basically I use the likelihood,
So you just
not the
have to
genotype.
for
Yeah.
the big one you do?
Up
Or
to
you use that one? I'm trying to understand what you use
Okay.
as the input.
Okay.
When you
For,
do this comparison, right? So like
I see. Okay, so let me describe it more clear. So for the bigger input, I have the sequencing rate, then compute the likelihood for every SNP. So likelihood to be 0, 1, or 2, right? So then this combined with the reference haplotype as the input for the big one input.
I see.
And for our method, I
You just
directly,
use the sequence data directly.
yeah, I just use sequence data directly, yeah. This paper has already been published, and we have the software online. So I think I'm done here for the project. And so finally, I want to make some acknowledgement to my committee here. So so there are a lot of things I want to say here. So first of all, I want to thank my advisor, Allele, for this amazing PhD time. I still remember the first year I came here, I almost have no knowledge about biology. And I mean, I asked a lot of stupid questions. Like, I remember I asked Jerry about like, what is the recombination? What is the genotype, haplotype, and the phenotype? What's these 3 types mean? And I literally asked this question. And just by help of Allele, I finally make this. I also like to thank Allele for allow me enough freedom to work on different problems that I'm interested in. And actually, I enjoy a lot for the first project, spatial ancestry analysis. I mean, I really enjoy that. I mean, if without Allele help, I probably cannot get that problem to work, and probably cannot even make this PhD. Yeah, I would like to thank Allele a lot for the amazing PhD. Second, I would like to thank Bogdan. Basically, I worked with Bogdan for my last year, from last November, I think, so until now. Actually, I physically moved from Allele lab to Bogdan lab. And I mean, it's really amazing staying Bogdan lab, because I can bug him all the time about different questions. So I think we probably have discussion almost every day about the projects, and I really learned a lot from the projects, from the discussion, especially in the population genetic stuff. Because my background is probably more computational, and for the population genetics, I don't know that much. And actually, this year I collaborated with Bogdan for many projects. It's probably be the most productive year in my. Also because it's my final year, so so it's very productive. Actually, I still have 2 more projects to go. not finished yet, but I'll try to finish it. Also, I would like to thank Wei and Jason for being a member in my committee. I mean, very, very unfortunately, I don't really have the chance to work with Wei and Jason. But I would like to thank Wei and Jason for your comments, especially in the oral qualification for those haplotype and population genetic stuff, such that I can make the final thesis work. Yeah, I would like to thank for you guys. Also, I want to thank all my collaborators in mostly in UCLA, and basically that's the photos in the lab retreat, because I work with the 2 labs. We have a lot of good time there, and I really enjoy the time in the 2 labs. Also, I would like to thank Aaron Halperin because the first project, spatial ancestry analysis, are like heavily collaborated with Aaron, and we probably Skype many times, even in the night, to discuss some problem. And I still remember Aaron gave some crazy math and helped a lot for the project. I would also like to thank John Mabry. He used to be in UCLA, but he moved to Chicago. Actually, I rotated with John in my first year, and actually that's the first time I get exposure to the population genetics, and then I become fascinated about that stuff. Yeah. Also, Dan, he is one of my collaborators. Used to be in our lab, now he's in IBM. Yeah. So, thanks for coming to my defense. I think I'm done here. So I'm happy to take any questions.
Any questions from outside the community? Yeah.
I have a question about the SPAMIX. How high do you go in the recombination rate?
Recombination rate?
Like the different recombination rate, or is it like a fixed one?
The recombination rate, we use because we have the SNP location, then we compute we assume a constant recombination rate, then we compute we assume the constant recombination rate between every location. Then we can compute the recombination probability between every 2 SNP using their distance.
Okay, so you just write that. Okay.
However, if you want, there we actually have the setting of different generations. So if you have more generation ago, the recombination will become higher. There is a linear relationship between the recombination probability and the generation.
And about that, the harsh, do you remember the illustration you showed? There was a set of H's, which is the copy of haplotype, 2 copies that you were reconstructing, and you had 2 different reference haplotype plots that every time you want to minimize your error correction, you will compare your reconstructed haplotype with them, right? Every time, and then you compute the error correction and try to minimize it. How do you actually select the very first initial haplotype?
Oh yeah. You mean, yeah, I understand. So the first, so I mean, the algorithm actually works when you randomly initialize that. It actually works just slow, so it might take like several hours until you get a better solution. So in reality, what I did is first I initialized the S and the H using the haplotype facing method. Then basically it's like a warm start of the whole algorithm. So you can do cold start, just slow, and warm start because the first you will get a roughly good solution. Then start from there, then become better. Yeah. Mm.
Okay, questions from the community? Yeah, so I have a couple questions. So one, with the Are you claiming you're like state of the art better than any existing imputation methods? Or what, so your baseline was sort of the model without additional stuff, but is your method the state of the art against all other imputation methods now?
So what the other imputation you mean from?
I mean, you're claiming you have a method to do genotype
I see.
imputation, right?
Mm-hmm.
It seems like that there's a lot of methods out there to
I see.
do that. So is your claim that you're now the state of the art in that problem?
Yeah, I mean, what we compare there is for the traditional model, it's actually just standard impute method. The capital impute. Because the capital impute is just like to find the two paths in the reference to get back the genotype. That's our baseline method. But however, there is basically we set the lambda to be 0 basically. Okay, maybe I can go back.
I mean, like, for example,
Mm-hmm.
when the 1,000 Genomes project releases an imputation of genotypes or something, what software are they using?
Oh, I don't use software there. But what I compare is because of, I need to show there. Yeah, here. So here, this baseline basically just represents lambda equal to 0 in our model.
Right, but I mean, you're not comparing to other software out there.
No, here I don't do that because in this project, I want to show conceptually, if you modify the hypertype calculus model in this way, you will get a better solution. But we didn't do extensive experiments to compare with other softwares.
Okay, so you don't know whether it's state of the art or not?
No, here we didn't compare with that.
Okay. Another question. So you also showed results for that your spatial method can help find gene number selection.
Mm-hmm.
Have you compared to other tests for
Yeah,
gene selection? And
yeah,
what's the overlap, and is one method better?
Yeah, we do. Yeah, in the paper, in the supplementary of the paper, we do compare with. I think we compare with a couple of them. One is the IHS, and the one is I think it's by Coop in the UC Davis. He has a method about the Bayesian-based environment correlated method. We also compare with FST. Yeah, so basically we have 3 methods to compare for the selection there, and we actually find basically we find that for the lactose region, every method can detect that, but for those FOXP2 and OCA2, only our method detects. Partially because our method is trying to detect the selection based on the gradient, but for FST, it just measures the divergence between populations. So it won't. I mean, basically if a SNP follows a slightly gradient, and the other method, if they want to compute the divergence between population, it won't show large divergence. But if you compute the gradient from that pattern, you might get a lot. So partially because this, and for some genes, only our method detects.
Final question. So, Trace, for this haplotype phasing, did you see the recent paper from Wen-Yun using high-C data as sort of a new strategy for doing haplotype phasing?
When is that published?
It was a few weeks ago in Nature Biotechnology.
I didn't read that yet.
Okay.
Yeah.
So if you had, I would be interested in your take on it.
So it's
that strategy can be combined with what you're doing.
I see. So it's a different read structure for that.
All right. So high-C allows you to find interacting locations. And the idea there is that it's finding interacting locations on the chromosomes, and that the assumption of their method, which they verified, is that if you're on
one chromosome, you're more likely to interact with yourself than the other alleles of the same chromosome. So when they find two interacting locations in the high C gate, you have SNPs for both of them, you know that you're likely to be on the same haplotype, and this can even give you longer range interactions.
Okay. Yeah, that one should, if we have the long range of, it might be equivalent to the very long read. Like, yeah, it should help for our method, I think. Yeah. But, yeah.
Any more questions? John, you have anything? Yeah, let's thank Wen-Yun one more time.
Thank you.
So there's a reception in our lab right now.
Yeah.
And I guess for everyone except for the committee, and Wen-Yun has to discuss. Okay.
Okay.